Hi, I'm Erika Kague from the laboratory of Shannon Fisher in the Department of Cell and Development Biology at University of Pennsylvania.
I'm Chris Weber, also from the Fisher Lab.
And I'm Shannon Fisher.
Today we'll show you a procedure for analyzing the function of enhancer elements using zebrafish transgenesis.
We use this procedure to study transcriptional regulations of genes important in skeletal development.
So let's get started.
To identify non-coding sequences surrounding candidate genes, we use the algorithm FastCons, which is accessible as a track on the UC-SC genome browser. We are primarily interested in genes expressed during skeletal development, but the same approach can be used to study any genes expressed in early development. In the example shown here, we have identified conserved sequences in the interval surrounding the human bumper gene. We examine the entire interval to the adjacent genes and determine a loci-specific LOD score cutoff corresponding to approximately the top 5% of non-coding sequence. After selecting sequences, design primers using Primer 3 to amplify each sequence from human genomic DNA. Primers should be selected to amplify the conserved region plus 30 or more base pairs on either side. Amplify the region of interest with high-quality polymerase using human genomic DNA. Use standard laboratory molecular biology procedures to clone the amplified product into an expression vector. The vector used in this protocol is PGWC-FOSGFP, a gateway destination vector. Following vector creation, Toll-2 transposase enzyme, required for transposition, is first transcribed in vitro using the M-message